Good afternoon, everybody. We are continuing on our mini series on molecular pathology for surgical pathologists. I'm Anna Plotkin, Director of CPD at the University of Toronto, and our presenters and keynote speakers, Dr. Yael Fisher, who presented last week with basics of molecular pathology, and hopefully everybody remembers everything, and Dr. Elan Hahn. And today I understand, Elan, you taking the stage.
Yeah.
Please, before you enter the room, mute your microphones because we have a high number of attendees. Thank you. Elan, the podium is yours.
All right. Thank you very much. Thanks for the introduction. I'm Elan. I'm a head and neck and molecular pathologist at Mount Sinai, and very happy to be speaking to you all today. Yael will be monitoring the chat, so if you have any questions, Dr. Fisher will answer them live. So we can get started, I think. So I have no relevant financial relationships to disclose. And we'll start with a bit of a review from what we talked about last time. And these slides are courtesy of Dr. Fisher. So we have DNA consists of 4 bases: adenine, thymine, guanine, and cytosine. And they are the complementary base pairs are held together by hydrogen bonds, A and T with 2 and G and C with 3. So having a bit of a stronger bonding between G and C is something to take note of, and we'll touch on it a little later in the talk. Really, when you have varying degrees of G and C relative to A and T, it can make things a little more difficult. The central dogma of DNA is that DNA is transcribed into RNA, which is then translated into protein, which will carry out the functions in the cell. Last week, we also covered copy number variation. And this is change in the number of copies of a segment of DNA. So these could be, even just parts of a gene, entire genes, or whole chromosomes. And we could look for this by FISH, which Dr. Fisher talked about. And we could also look for it by next generation sequencing or microarray, different techniques where we could be seeing, how much of a certain region of DNA there is. Next, Dr. Fisher went on to cover gene fusions. And this is when there's a breakpoint in 2 different genes that then join up to make a functional product. We also spoke about how a lot of these breakpoints are located in introns, which are quite large and difficult to sequence across, which is why, DNA sequencing for fusions is not as good or not as easy to do as RNA sequencing for fusions, where we cut out the introns and are just looking at exons, which is much more manageable. Additionally, RNA, there's an element of expression that goes into the production of RNA. So when a fusion transcript is expressed highly by a cell, we will see more of those fusion reads as well. Next, Dr. Fisher covered single nucleotide variation and little small deletions insertions. So this is a change of, 1 base or a few bases that will either, make a new amino acid, make the same amino acid, cause a stop codon, or cause a frame shift that will then lead to premature truncation, typically. So that's a good speedy review of what we talked about last time. So now moving on to the learning objectives for today. We're going to talk about the typical flow of a specimen through the molecular lab. So after today, I think we'll have a much better understanding of that. In addition to pre-analytic quality factors to be aware of, the basics of nucleic acid extraction, different molecular assays. So what do we do with that nucleic acid? Specifically looking into next generation sequencing approaches and how to choose the correct assay. So we'll start off with specimen selection, move on to extraction, different molecular methods, including PCR, Sanger, and next generation sequencing. So first we'll start off with specimen selection. So before a specimen comes to us, there are important things to be aware of. And this is really that first step in the specimen journey. So it's removed from the patient, and then it comes across our desk. There are some specimens that directly will go to the molecular lab, but as this course is targeted towards surgical pathologists, the specimens that we're talking about will come across our desk, will come across our microscope. And there are certain things that will make it, either more likely to work or more ideal as a choice if there are options for molecular testing. And there are certain things that make it less optimal. So cold ischemic time is something that would make it less optimal if it's too long. Fixative use is important to be aware of, decalcifying solutions specifically. Proper sectioning for fixative penetration. This is typically in larger specimens like, a leiomyomatous uterus, for example. Fixation time. It was specifically an issue, I think, around the pandemic where there were grossing delays, where specimens were spending too long in formalin. Otherwise, there are certain contaminants that could be present in the tissue, like melanin, mucin, collagen, or necrosis. And a separate issue to be aware of is tumor cellularity. So how many tumor cells do we see relative to other cells? And specifically lymphocytes tend to be a common, contaminating cell type, or stroma and stromal components. So moving on to fixatives. Neutral buffered formalin and alcohol are good. Unbuffered formalin, buins, heavy metals, mineral or organic decalcifying acids are not good. So some of the formalin, extended time in formalin will cause cross-linking or deamination artifact. Acids will cause depurination. It will modify the bases and fragment the DNA. And one of the methods it does that is by hydrolysis. Moving on to the topic of cellularity. When we talk about tumor cellularity in molecular testing, we're talking about the percentage of nuclei that are tumor nuclei. So it's the tumor nuclei divided by the total number of nuclei in the selected area. And, area is one thing, and you want there to be enough number of cells. The raw number of cells, you want there to be enough. But more important than that is the cellularity. And how many of those cells are actually tumor. So just to start off with a question, which specimen or specimens, there may be more than one correct answer, are suitable for molecular testing? And this is keeping in mind, let's say this hypothetical assay requires a tumor cellularity of 30%. So I'll let some people answer in the chat. Okay, so I'm seeing a lot of A's, some C's, some B's, but mostly A's. And that's right. So when you have a tumor cellularity percentage of 30%, A would be the only one that's suitable. B, although it looks like there's a lot of tumor, there's a lot of necrosis as well. So this wouldn't be a suitable choice for testing. And C, there appear to be some tumor cells, but the vast majority of nuclei in that area are lymphocytes. And even though these lymphocytes are smaller, they have the same amounts of nucleic acid as tumor cells. So they could really drown out a signal. So if we go forward and say we have more of a modern type of assay, which specimens are suitable for molecular testing if cellularity is 20% as a requirement? So by default, we know A is right, but are any others going to sneak in? So I think B is still going to be there's too much necrosis. So again, I don't think it is a suitable specimen. C is going to be far lower than 20%. D is going to be close to 20%. So here, this is a scenario where, number of cells might be on the lower end, but the percentage of tumor cells relative to other cells will be adequate. So I think that would be an appropriate choice. And, this may take, submitting additional slides so that we get more number of cells, but the percentage will be good enough. And this factors in downstream when we're looking at mutations or we're looking for things at a certain allele frequency. And if we don't have appropriate cellularity, we won't be able to find it at a required allele frequency. So there are ways we enrich these slides or these specimens for testing. And how do we enrich them for tumor? So certain techniques have no enrichment, like cutting thick scrolls, scraping unstained slides entirely. There's macro dissection, which I think most labs are using currently, where we circle an H&E and then using the naked eye, we could scrape off the tumor within the circled area. Manual micro dissection using a light microscope, which is what we use here after circling to make sure you're getting the circled area scraped for extraction. And laser capture microdissection, which is a laser coupled microscope to capture the tissue of interest. So depending on your lab's capabilities and the requirements of the test and the tissue that you're using, you may use different techniques for enrichment. So certain quality control measures can be implemented to help at this stage of specimen selection. And I think ensuring tumor specimens are put in formalin immediately and properly sectioned for penetration is very important to prevent autolysis. These cases where there may be molecular testing as a requirement should be prioritized for grossing to avoid overfixation as close to 48 hours as possible. And separating course of biopsy to preserve tissue for IHC and molecular will also help ensure that there's enough tissue and enough tumor cells for testing. So now I'd like to move to a case that we saw here. So this is metastatic appendiceal mucinous adenocarcinoma. It was tested on our NGS panel with a limit of detection of 5% allele frequency. And this is what we saw. This is, as Dr. Fisher showed last time, this is called the integrated genomic viewer. It's how we represent sequencing data, raw sequencing data after it's been aligned to the reference genome. And I'll just point out a few of the features here. So this is the reference genome. These are the bases in the reference genome. Here, there's a protein domain track, which will tell us what domain of the gene, what protein domain of the gene we're looking at. And this here is a measure of coverage. So these are actually vertical gray bars that are all blending together. And, gray bars of a certain height mean it's covered to a certain amount. And as you can see here, selecting our region of interest, we covered this region 679 times. And as you see, each of these gray bars that are horizontal is one unique read. And wherever it matches the reference genome, there is no colored base. Wherever it's different than the reference genome, we have a colored base, and it will show how it's different. So in 15 instances, this was A, which corresponds to KRAS G12V, which is very important in treating colorectal carcinoma. But as you can see, the allele frequency is 2%. It's lower than our limit of detection. So my question is, what's your next step? And again, I'm going to preface that all of my questions have more than one right answer, potentially. Would you report this as negative? Would you repeat the test? Would you check the block, check the slides to see what the tumor cellularity was, if you don't remember, at the time of circling, or if somebody else circled the test, or would you look for another sample? So I got lots of C's, some D's, I'll let the answers come in for just a touch longer. Okay, so it looks like a mix of C's and D's. I think that is true. And I think if you would go with looking for another sample, then repeating the test if you found a better sample would be a good way to go. For this specimen, we didn't have any other samples, so we couldn't repeat the test, or all the other samples had similar cellularity. So checking the cellularity, you could see the area that was circled, trying to get rid of, some lymphocytes, as you can see at the periphery, if you could see through the marker. There's some lymphocyte aggregate here that could really dampen the allele frequency, especially in a case with such low tumor cellularity. So you can see it's mostly mucin, which happens to inhibit PCR as well. And there's just scant strips of tumor epithelium. This was the other part of the same slide that was circled as well. Again, trying to get rid of these lymphoid aggregates that are, towards the periphery of this met, not really being able to avoid them all, unfortunately, while still wanting to get enough tumor cells, which is so scanty. So again, another question, what's your next step? Reported as negative. It was lower than our lower limit of detection. Repeat the test. Report as positive with a comment or look for another sample. In this case, I think I've said that, B and D don't really apply because there were no other better samples. Although we could repeat it with the same, try to maybe circle a little less and see what we get. Or are we confident with our circling? So I see some A's and mostly C's. So what we did is we reported it with a comment. It does fit the tumor cellularity that we saw. So seeing this mutation at a low allele frequency, I think, we're trying to do our best to give the clinicians an answer here. And it really fits with the appreciated cellularity. With, let's say this were a very cellular tumor, finding a KRAS G12V at 2%, I wouldn't trust that. But seeing as this correlates with the appreciated tumor cellularity, there are no other specimens of better cellularity, and I don't think circling a smaller area would really give us more success. We felt it was appropriate to report it in this context with this comment. So next, we'll move on to extraction. So once the area is circled and scraped, then comes extracting nucleic acid from that sample. So just to review, again, each strand of DNA is built through phosphodiester bonds, and those two strands are held together by hydrogen bonds. RNA is single-stranded, and it has a hydroxyl group, this OH, compared to the H of deoxy, no oxygen, ribose in DNA. So there are important considerations before we start extraction. One is the sample type and collection method. How was the sample collected? Is it an FFPE? Is it a cytology specimen? Nucleic acid needed. Do we need DNA or do we need RNA? RNA tends to be less stable than DNA and is more susceptible to degradation and RNases. How much nucleic acid do we need? And what quality parameters are we going to be looking into to see the quality of our extracted nucleic acid? Extraction has 5 steps. Lysis, breaking the cells, cleanup, getting rid of contaminating material, purification, getting just the nucleic acid, washing away unwanted material, and elution, freeing up our nucleic acid from what we're using to purify it. And we'll go into each of these steps in more detail now. So lysis, we're trying to break the cells to release the nucleic acid into the solution. You could use various methods alone or in combination together. It could be done physically with grinding or sonication, enzymatically, an example of which is proteinase K, or chemically with detergents or pleiotrops. Then we want to clean up, get rid of all these cellular debris, the proteins, the lipids, and we could centrifuge, we could filter, or we could use beads to help with that. Purification, just getting the nucleic acid. We could use solid phase purification, which incorporates binding matrices like columns or magnetic beads. These could be silica chemistry, cellulose, or ion exchange. And the liquid phase would be solution-based, like in alcohol precipitation. So two of the more common purification methods are spin columns and magnetic beads. So first, looking into spin columns. These lye samples are transferred to a spin column within a microcentrifuge tube, and then you centrifuge or vacuum, causing the nucleic acid to bind to the column while the waste passes through. And then you'll see steps 3, 4, and 5 where we wash away the waste and elute the nucleic acid off of the column. This is an example of a silica spin column. And here, a similar principle is by beads. So after lysis, you add magnetic beads. Then you wash away the unbound substances, which would be the contaminants. Then you elute the nucleic acid from the magnets, and that's what you use. So washing, use buffers or alcohols to remove unwanted materials like protein, salts, lipids, other contaminants. And elutions, you're trying to free nucleic acid from that binding material. Nucleic acid is soluble in low ionic strength solution, so we could use nuclease-free water or low salt buffers. So now that we have our extracted nucleic acid and it's purified, we want to look into this purity. So one way to look into this purity is by absorbance spectrophotometry. And there are absorbance differences between nucleic acids and proteins, and we leverage that to look at the ratio between wavelengths of absorbance. And we look at the wavelength for nucleic acid, which is 260, relative to the wavelength for proteins, which is 280. Another important aspect of the quality of extracted material is quantity. So we could use fluorometers, fluorescent dyes that selectively bind to nucleic acid, and the fluorescent intensity will correspond to the nucleic acid quantity. Even if there are contaminants there, we'll still get an idea of the quantity of nucleic acid. Next is integrity. So yes, we have nucleic acid, but of what quality is it? How long are the fragments? And is there a lot of fragmentation? So electrophoresis is something that you could use, and as you can see, degraded nucleic acid smears. You have multiple fragments of all different small sizes, whereas intact or better more DNA with better integrity will show strong bands of higher molecular weight. RNA, we use the RIN, the RNA integrity number, sort of an automated capillary electrophoresis system, or you could use the DV200, the percentage of RNA fragments that are greater than 200 nucleotides. And smearing or low RIN or DV200 values suggest degradation or inhibitor presence. So that's another important aspect of quality of extracted material to look into. And there are some platforms where you can actually use PCR quantification for quality assessment. So this is a quantitative PCR method, which we'll talk about later in more detail, what qPCR is. And it assesses both the quantity and integrity of genomic DNA by amplifying specific target sequences of different lengths. Cybergreen is an intercalating agent that gives off fluorescence, and it intercalates into double-stranded DNA during PCR. And the fluorescence will increase when the amount of amplified product increases. So that allows for real-time quantification. And again, we're going to talk about this real-time qPCR a little later as well. So by comparing the amplification of a short and long amplicon, the assay will provide a degradation score, which will reflect the DNA integrity. And as you can see, the top figure shows high quality human genomic DNA, which is the HG, where we have a good amount of long fragment DNA here. So you can see our samples in gray, and it's amplifying to a detectable product early on, only after whatever 17 cycles, for example. Whereas our damaged DNA only amplifies to a detectable product after 30 cycles, which is more non-specific to get amplification at that point. So this helps us say we have a lot of, small fragments, longer fragments, and even longer fragments. So once we have this extracted nucleic acid, it's time for storage. So we could store it for more than 7 years at minus 80 degrees Celsius, less than 7 years at minus 20 degrees Celsius, and less than 3 years not really recommended at 2 to 8 degrees Celsius. So now we have our extracted nucleic acid. The next step in the workflow is to go to, different molecular techniques that we're going to use. So first, we'll start by discussing polymerase chain reaction, PCR. So PCR is an enzymatic method that amplifies double-stranded DNA, resulting in fragments of the same size and identical sequence. And it's multiple cycles of the same thing: denaturation, annealing, and extension. And we'll talk about that. The components of PCR are magnesium chloride, which is a cofactor for DNA polymerase, buffer, which maintains a stable environment, DNTPs, which are the building blocks of the new strands of DNA polymerase, which is the enzyme responsible for amplifying and extending these strands, and our target DNA, which is what we intend to be amplifying. So first, denaturation. Double-stranded DNA is denatured into a single-stranded DNA by heating the reaction to 94 degrees Celsius. Next, we have annealing. So our primers, which are short oligonucleotides, defining the region of interest that we want to amplify, these are allowed to anneal to their complementary sites in our target DNA and our template by cooling the sample to 50 to 60 degrees. So these primers are very targeted at specific areas of interest. And once we cool it down, they can actually bind to the DNA that's there of our sample in order to begin the next step, which is extension. In the third step of PCR, polymerase is activated by heating the sample to 72 degrees and allowing it to begin synthesis of a complementary fragment of DNA using the sample DNA as the template. And after multiple cycles, we go on to exponential amplification. As you can see, once we're past the fourth cycle at 32 copies, and if we get to 36th cycle, we're going to be making 68 billion copies. So this leads to exponential amplification of targeted regions. And once we have our amplified product, we could, detect it in a number of different ways. One of those ways would be Sanger sequencing, which we will move on to next, but sequence our product. We could detect size differences in the product, either by electrophoresis or restriction enzyme, of a specific site of interest in our target that will then create, fragments of known length. Or we could look for the amount of product, and that's quantitative PCR. And that's similar to what we described in that last quality method for evaluating extracted nucleic acid. There are multiple different PCR assays, and we're going to talk about qPCR or real-time PCR, but there are other assays as well: nested, multiplexed, reverse transcription, or long-range PCR, and then qPCR. So qPCR or quantitative PCR, it allows for rapid identification of a target. It eliminates the need for post-PCR processing because we're getting a real-time value. It's specific for the area of interest. You can multiplex it and look for different targets. It tells us how much there is. That's in the name. It's quantitative. And it uses dyes or probes. So what we do with real-time or quantitative PCR is you detect, by using a dye or a probe, the amount of amplified product. And you see that CT value is the threshold value where we determine that there's an identifiable product. So the range of CT values that's specific is typically 15 to 35. A CT value less than 15, it's not really considered to be within the baseline phase and does not reach the fluorescence threshold. Ideally, there's a linear relationship between the CT value and the logarithm of the initial copy number of the template, which is the standard curve. And using the standard curve when the amplification efficiency is 100%, the CT value for quantifying a single copy of the gene is usually around 35. So anything above 35 for a CT value, theoretically, the initial copy of the template is less than 1, and it's not really considered statistically significant. So when we look at a qPCR plot, we're seeing at what cycle did we get detectable amplified product. The earlier we get it, the more of our target there is. The later we get it, the less of our target there is. So next, we'll move on to Sanger sequencing. Sanger sequencing, first-generation sequencing, allows us to determine the order of bases in our nucleic acid. It works by elongating this chain by one nucleotide at a time. The pool of incorporating nucleotides is comprised of dideoxynucleotides, which actually terminate extension. And we're going to go into this a little in a couple of slides as to why this is so important. The OH group is necessary for elongation. So if it's lacking, replication or elongation stops. The components of Sanger sequencing include our template DNA, primer, DNA polymerase, our buffer, again, to stabilize everything, deoxynucleoside triphosphates, and DDNTPs, dideoxynucleoside triphosphates, each with a different fluorescent label and a much smaller molar concentration than the DNTPs. And because we're looking for a very specific region of interest, once we what we get is, and we have a much higher concentration of DNTPs relative to DDNTPs, what we get is amplification of basically one step at a time, all the way up to the complete sequence. And each of these terminating strands will have one of those fluorescently labeled DDNTPs at the end of it. So we're generating fragments of different lengths, each labeled with a terminating fluorescently labeled DDNTP. And by running these on electrophoresis, we're able to determine the sequence. These electropherograms, you could either run it on a gel, or you could generate an electropherogram and convert that to sequence data. So what we see here is evenly distributed peaks, each corresponding to a certain base call in a certain position. And these are separated by size. And I'll go through a few examples here. This is an example of KRAS mutations in colorectal carcinoma. And I pulled this from a paper where they were looking at these different mutations, and we could go through this together. So this is the reference wild type sequence, and you can see the arrow is pointing to where we're going to have a mutation on the left side. So starting at the top, instead of a G in this position, we have an A. So you see that this is heterozygous. We have a little bit of a bump that's black, that's showing a G, and we have a bump that's showing an A. as well. So this is at position 436, we have a G that's going to an A, and that corresponds to KRAS A146T. In our next example, we're looking at codon amino acid 134, and we have a change from a C, again blue, to a T, red. And again, this is heterozygous. In a proto-oncogene, we expect heterozygous changes in the absence of copy number changes. Here we again, instead of a G, we have an A, a small little green bump. Here, at a position 150, we have a stop codon that comes in here, and instead of a C, we have a T and a C. And again here, instead of a G, one black bump, we have a black and a green. So an A. And here, instead of an A, we have a G as well. So all of these are examples of what it looks like to have a missense variance or a substitution at a single position by Sanger sequencing. Here is, this is a condition called Dowling-Dego's disease, and it's an analysis of the specific gene POFUT1 gene family. I chose this example to show what a one base pair deletion looks like by Sanger sequencing. So this is the unaffected individual. Essentially, this is the reference for this gene, for this area. And as you can see, it goes GAGTACTT, CCC, ACTGA. And if we look here, everything's fine until the T, until the G, until the A. But then once you get to this G, you have an overlap of a T. And then what should have been a T, you have an overlap of an A. And what should have been an A, you have an overlap of a C. So what happened is you just deleted this G. And then everything shifted because the next, instead of the G, the next it was a T. So as you can see, you have an overlap of the reference sequence with the reference sequence starting one base pair later. So you have a G overlapped with then this next exact sequence, and that's what a deletion looks like of one base pair in Sanger sequencing. If it would be more than one base pair, again, you'd have just superimposed the downstream sequence on where the deletion starts. So Sanger sequencing has known limitations. It has a relatively low sensitivity when we're comparing it to next generation sequencing, where we need an allele frequency of 20%. This is hard in cases where there's low tumor cellularity, where there's tumor heterogeneity, there's low quality target nucleic acid. So all of these reasons make the choice of Sanger sequencing in those cases suboptimal. Quality tends to degrade after 700 bases, and there's poor quality close to the primers. So primer selection for areas of interest is very important. And you're really only sequencing your targets very specifically. So after talking about Sanger sequencing, we could move our discussion to next generation sequencing. This is also called massively parallel sequencing. Well, we're basically doing Sanger sequencing massively parallel, all at all at once. So multiple simultaneous reactions with data collection in real time. There are different platforms, including Illumina and Ion Torrent. And we're going to talk about each of these and their differences. So the general NGS workflow is library preparation. We're preparing our template that we're going to sequence. Amplification. We're amplifying our targets. Reaction and data collection. So doing the reaction, actually carrying it out and collecting the data from the sequencing in real time. And then since we're creating so much data, bioinformatic analysis to make sense of it. And we're going to get to that in our next session. The unique combination and specific protocols and chemistries is what distinguishes one platform from another. In general, library preparation starts with shearing nucleic acid, usually by sonication, into fragments, 100 to 500 base pairs in size. enriching for targets, and this will differ by different platforms. We'll get there. Ligation of an oligonucleotide barcode to our fragments, and these will have a specific sample ID because we're going to pool multiple samples for the reaction. And having this ligated oligonucleotide barcode will help separate the reads bioinformatically for each sample after sequencing. And also ligation of oligonucleotide adapters for PCR amplification that are going to help for either platform to actually carry out the reaction. And this is what our fragments will look like. The fragment in green, our barcode in orange, and then adapters on either end to help carry out the reaction. So starting with target enrichment. For Illumina, regions of interest are isolated by hybridization to a probe. So we're specifically looking at probes that are going to be complementary to regions of interest, and they will hybridize to our template DNA and isolate it that way, and that will enrich for our targets. Ion Torrent is amplicon-based, and we have PCR primers for a region of interest. We're going to amplify our template to create a lot of reads of those regions of interest, which will make it detectable. So we call Illumina's chemistry capture-based or hybrid capture, and Ion Torrent's chemistry amplification-based or PCR-based or amplicon chemistry. And generally, amplification-based assays, target enrichment is PCR amplification of the desired region, as we mentioned, using primers, as you can see in the top left of the figure. A tiled amplicon approach is depicted where we design primers to generate multiple overlapping amplicons of the same region to avoid dropout of alleles, and we'll talk about that later. The sequencing reads generated will have the same start and stop coordinates because it's based on our amplified product by PCR. In hybridization capture-based assays, target enrichment is based on using biotinylated oligonucleotide probes complementary to our region of interest. As you can see on the right side of the figure here, the probes hybridize to target regions contained within larger fragments of DNA. As a result, these flanking regions, as you can see here, are also captured and sequenced. And targeted fragments are isolated using streptavidin magnetic beads, followed by washing, elution amplification, and sequencing. And we'll get into that as well. The sequencing reads from these molecules will have unique start and stop coordinates. So they're not all going to be identical, like amplicon-based assays. And they'll be aligned to the reference and allow identification and removal of PCR duplicates where it looks like maybe we amplified the same read or the same hybrid as the same captured fragment more than once. In bioinformatic aspect of the analysis, that's something that can be filtered out. So the amplification for Illumina is by bridge or solid phase, and we'll get into that. And emulsion is the technique that Ion Torrent platforms use. So what we're trying to do is generate a spatially separated lawn, is what it's called, of amplified product, so that we could create clear and strong signals in our sequencing reaction. So looking at the Illumina hybrid capture-based assay, this is performed in a flow cell. It's a flat glass surface with covalently attached primers that are complementary to ligated adapters. So anything that we've now pulled down through our capture process has adapters on it, and they will bind to their complementary primers that are affixed to this glass surface. So the newly synthesized strand here is covalently attached to the flow cell surface. The hybridized strand melts away. Then what we have is this strand flips over to hybridize to the adjacent primer, forming a bridge. Now, polymerases extend the newly hybridized primer, forming a double-stranded bridge. The double-stranded bridge is denatured, and now you have two surface anchored copies of a covalently bound single-stranded template. This has created both forward and reverse strands at this point. So the reverse strands are cleaved away and washed away, leaving an entire cluster of only forward strands. So then we apply nucleic acid at the appropriate concentration so that we ensure spatial separation. So we can't have too much nucleic acid from any one specific sample. Otherwise, these clusters will be too close together. And when we are reading the fluorescent images later on, they'll be indistinguishable. So what happens next is fluorescent labeled modified nucleotides are added together all at once with reversible change termination. And it's repeated cycles of adding a nucleotide, detecting the fluorescence, taking a snapshot, then removing the fluorophore and doing it again. And so then we're able to see the elongating sequence by subsequent images that are taken. And then this whole process is repeated again for the reverse strands. And all of this data of colors at certain time points, at certain lengths of strand, is converted into base calls by the software and bioinformatic analysis. For ion torrent, we amplify through emulsion. So we have beads that are coated with primers that are complementary to our adapters that we added on. The DNA library and beads are mixed just right at a concentration where these droplets contain one piece of DNA, one bead, and they'll have all the ingredients for a PCR in there as well. And this is an oil immersion, so tiny, tiny droplets, which have polymerases, DNTPs, and template DNA and our bead. And you need a single fragment per droplet, which is why the emulsion process is so necessary. If not, you'll have polyclonal amplifications that will have mixed sequencing results. And what happens is we're going to separate these beads, these droplets of individual PCR reactions into microreactors. So, having PCR at this stage can introduce sequence errors or preferentially amplify certain areas which may skew our fragment representation. So basically what you end up with after this PCR is beads coated in 3 dimensions with amplified identical product. So what happens is each of these beads is then contained in a single well across the sequencing chip. And every time a nucleotide, a DNTP is incorporated, it releases a hydrogen ion. And this decrease in pH is detected here by the sensor, and that's interpreted into bioinformatic data. And how it works is they add one DNTP at a time that flows across the well. And if nothing's detected, then there's no signal. If something's detected, then we know in this specific well that whatever DNTP was being flown across is the one that's next in that sequence. And if there are multiple in a row, then they'll get a bigger change in pH. And this change in pH is linear up to 5. So if there are a repeat of 5 bases, the same bases in a row, then we start to lose sensitivity in detecting if it's 4, 5, 6, 7, or 8 A's or G's in a row. It's not as accurate. And that, having multiple of the same nucleotide in a row, is called a homopolymer, and it's a common source of error or artifact in amplicon-based technologies. specifically with the Ion Torrent platform. So comparing hybrid capture and amplicon-based sequencing, there's in hybrid capture a pro, which I've colored in green. There's no introduction of PCR-related sequencing errors. You do need a higher amount of starting material. A benefit is capture probes are significantly longer than the PCR primers and can therefore tolerate the presence of more mismatches in the probe binding site without interfering with hybridization to the target region. This will avoid allele dropout, which can be seen in amplification-based methods, where you really need a better match because these primers for the amplicon-based methods are shorter. It's more scalable with larger machines. You can do more samples at once. It has a longer runtime. But a good benefit is neighboring regions are interrogated, where we could find novel fusion partners and neighboring sequence changes that we may not see or that we wouldn't necessarily see in amplicon-based technologies. So amplicon-based, since we're starting off with a PCR off the bat, you could start with limited starting material. Again, these short primers may lead to allele dropout because they can't tolerate much mismatch with the target. It's less effective in GC-rich regions, which have a tighter bond, and that impacts PCR. And that goes back to what we talked about at the beginning of the session, where G and C between them have 3 hydrogen bonds. So if you have a lot of G's and C's in an area, it may not amplify as well under the same conditions that an AT-rich area would be or a more mixed area would be. At the end of amplicons, there's worse quality. You could introduce sequencing errors that then get amplified, and that could also skew fragment representation. It has a shorter runtime, and it's much less costly, which is also important. So tiling amplicons, which we talked about, having redundancy and coverage of the primer and areas that are close to the primer or end of the what you'd expect to be amplified, is helpful for some of these issues. So we could go back to a case. This is a parotid lesion. As you can see, multinodular growth, somewhat well circumscribed within the parotid gland, lots of myoepithelial cells, quite monomorphic, with very focal duct formation. So the differential diagnosis was pleomorphic adenoma versus other entities that we wouldn't want to miss. So this was tested on an amplicon-based NGS panel that has both PLAG1 and HMGA2, in addition to many common partners. And sequencing was negative. What's your next step? Do you report it as negative? Do you repeat the test, repeat with a capture-based panel, or do you want to try FISH to look for break-aparts of these genes? Okay, so I see a lot of Ds and Cs. And again, as I said, there are multiple right answers for a lot of these questions. So D is definitely a right answer. We don't have this FISH available, so I elected C, which is another right answer. So amplicon-based methods for fusions, you need primers for both of the partners. So not just HMGA2 or PLAG1, but also the fusion partner. And if you don't have that, you're not going to detect the fusion. But once we repeated with a capture-based panel, where all you need is one of the genes, and then you sequence across the breakpoint to find the partner, you could find novel fusions. So this is again an IGV look at what a fusion would look like in IGV. So this is ST3-Gal5, and this is what was called as the fusion partner for PLAG1. So as you can see, we end right at the exon, and then we pick up with TAA, CC, GG. And if you look here in PLAG1, the beginning of the area that we're getting as part of the fusion, it has that same exact sequence. So our bioinformatic analysis, our fusion caller, called 146 reads of this fusion, because it had areas that mapped to two different genes, and that's what the fusion caller bases its calls off of. And what we have is the fusion of exon 1 of ST3-gal5 to the 5' UTR of PLAG1, which increased its expression. So that helped confirm the diagnosis of pleomorphic adenoma in that case. And I think this brings us to a nice spot where we could talk about sequencing with RNA or sequencing with DNA. So RNA must be converted to complementary DNA with the first transcriptase, and then we use that complementary DNA for sequencing. Once we have extracted DNA or RNA, we could either use RNAs or DNAs to remove the other one, the one that we're not interested in sequencing, depending on the target material. So RNA-seq is helpful for fusion detection because of what we mentioned previously. The breakpoints tend to happen at variable locations in introns, which are very long and hard to cover. But the fusion gene is transcribed into mRNA with just exons, which is much more reasonable and covered in a much easier way with high confidence. Additionally, some of these fusion reads or some of these fusion products, a lot of them depend on differential expression, where you're taking a gene, gene A in the fusion product that is driving expression of gene B, which is typically not highly expressed. So if you're just looking at DNA, you may not see, an increase in number of reads that will have the fusion. But when you're looking at RNA, which is governed by expression changes, this may help selectively sequence fragments of RNA that have the fusion in it. So RNA-seq is definitely valuable and used most commonly for fusion detection. In other cases, it's used for splice variant evaluation, where there's a splice variant somewhere close to the junction and we don't know how it's going to affect the DNA. We don't know how it's going to affect splicing. And a functional way to look at that is RNA. So do we see that this exon is actually skipped over or not? And that's similar to the MET exon 14 example that Dr. Fisher spoke about the other day. Another way we could look at RNA data is by gene expression. So a highly expressed gene will have more reads or will be covered more in RNA. And this isn't used clinically, typically it's not used clinically. There are some assays like, thyroid cytology, NGS predictors, which actually incorporate gene expression changes into their analysis. But otherwise, in general tumor sequencing, it's not typically used. So still speaking about rearrangements, there are different ways to look for rearrangements. We could use karyotype to look for, whole chromosome arms that are translocated to different chromosomes. Which has limited resolution. We could use FISH. You need to specifically know your target to have a FISH probe for that target and to evaluate it that way. And this could be done in a number of different ways, where you have a probe that covers the whole gene, and you're looking for that probe to be broken apart to indicate that that gene has been translocated. Or you could have probes for two different genes that then you see those two probes that are normally spatially far apart in the cell come together to show you a fusion signal. RT-PCR, which is reverse transcription PCR, you could do to look for fusions, but you would have to know both partners to amplify the fusion product. So you'd have one primer for gene A and one primer for gene B, and you'd sequence and hope to go across the fusion junction. And otherwise, it's NGS, and that's what we spoke about with RNA-seq. And you can also use DNA-seq or whole genome sequencing where you have better coverage of introns, but this is not without its challenges, both in finding a high confidence call in an intron and expression changes. So this brings us to another case. Another parotid tumor. It's happened to look like it's in a lymph node in the parotid. As you can see, there are mucus cells and some squamoid cells. And the question here this was sent for molecular testing. Can you identify a mammal-2 fusion? So first question, the specimen comes to you in the workflow. How will you enrich for the tumor? So we have lots of C's and D's, and that's right. Either one, some sort of targeted enrichment would be helpful in this case. So macrodissection or microdissection, maybe microdissection because tumor cellularity is even in the tumor areas is lower, but we have so many contaminating lymphocytes that all of our reads are going to go to lymphocytes if we use scrolls. So this came, and this actually had scrolls, and we didn't find any gene fusions. So what's the next step? Report it as negative, repeat the test, repeat it with this, enriched slides for RNA-seq, or try a different assay altogether. So we got C's and D's, and I think, again, those are both correct. In this case, we chose to go for fish because it was more cost effective, and even in the tumor area, there weren't so many cells, and CRTC1 or 2 mammal-2 fusion tends to not be that highly expressed. So I thought there was a chance we would miss it regardless. But either of those answers are correct. And fish was positive for mammal-2 rearrangement. So lastly, I want to speak about targeted versus unbiased platforms. So commercial panels or custom panels, those are targeted platforms. We have targeted panels here at Mount Sinai. There are other commercial panels that are available that are targeted. And these could either be amplicon or capture-based. You can have both that are targeted, as we saw. And for general purposes and general needs, these are enough. We cover the CCO or Ontario Health panel suggested genes that we're looking for, and you could make these panels quite large actually, and definitely have enough coverage to answer our clinical questions. Unbiased platforms like whole exome sequencing, whole transcriptome sequencing, or whole genome sequencing are helpful if we don't really know what we're looking for, and may become more cost effective depending on how many different types of tumors you're sequencing on your panel. Do you want to have different panels for each one, or do you want to have one test that you use for all of your specimens, and what the nature of your specimens is? Are you looking for novel fusions all the time or not? Or novel changes all the time or not? And that's something that may push you towards an unbiased platform. Whole exome sequencing, refers to DNA sequencing of all of the coding exons in the genome. Whole transcriptome sequencing is RNA sequencing of those coding exons, and whole genome sequencing is everything. So with that, I finished what I wanted to cover today, and like to open it up to any questions. And thank you all for attending and for joining us in this course.
I think Yael was answering the question as we go. Thanks, Elan, for this wonderful presentation. So if you have any questions, please type them in the chat, and Elan would address the questions.
So I see there's a question about the cost of some of the techniques.
Oh, yeah. So I can't give the exact dollar costs of the different techniques, but Sanger and PCR are much more cost effective than next generation sequencing techniques, which will be more expensive. I don't know if you wanted to weigh in.
Yeah, I think I can add that maybe it really depends on what you're looking for. So if you are looking for a specific fusion that at least one of the partners, then maybe fish would be more cost effective. But if you're not sure about that, or about the exact gene partners, you'll have to do multiple fish testing, and then NGS would probably be more cost effective. So that really depends on what you're looking for.
Mm-hmm. Agreed. And I see there's a question about extractions, core puncture of the block, or scraping. Both, as long as you're getting enough input, both can be effective. I would say with scraping of, marked slides, you may get, better enrichment for tumor specifically, where if you're going too far into the block, you may not be necessarily following the levels through. But both could be effective and used. I also see, will there be a course on using IGV and interpreting raw sequencing data? I'm not sure if we're going to do it in this session or, but I think we'll probably touch on that in the bioinformatics lecture next week. Is that right?
Yeah, I will try to expand some more about that. I'm not sure it will be a whole course, but I'll try to expand some more.
There is one more question. Do you use a frosted glass or charged slides to scrape the tumor?
I don't think it matters.
And you will have a lot of things, including Yael, that I'm not sure Yael would be able coming. Thank you coming from you automatically. Okay, with that, would like to finish, and thanks everybody. We'll see you next week, Tuesday at the same time. Thanks Elan again, and thanks Yael for this wonderful presentation. Bye bye, everybody.
Thanks.